Good morning and welcome to all. Today we are going to discuss about the protoplast and sphaeroplast. So this protoplast and sphaeroplast are, both of them are altered forms of bacteria or yeast, in which the principal shape maintaining structure of the bacteria is weakened. What is the principal shape maintaining structure of the bacteria? It is nothing but peptidoglycan within the cell wall. It is the shape maintaining structure of the bacteria. It is weakened in both protoplast and sphaeroplast. This may result in the formation of a sphere-like structures. Each bacterium forms a sphere, which is the shape that allows the bacteria to withstand the rigors, particularly osmotic, of the fluid in which it resides. Actually, the cell wall is the defensive stress-resistant layer of the cell, and the cell loses this defensive stress-resistant layer, that is cell wall, which is a protective layer, and the cell loses this protective shield in its absence. Higher concentration, what happens? How this protoplast and sphaeroplast generate? We can discuss. If the higher concentration of solution is outside the bacterial cell, there is a tendency of the solution to move from higher concentration to the lower concentration. So what happens? From the outside, the solution enters inside the bacterial cell. This may result in swelling and eventual cellular bursting. Conversely, whenever there is an increased concentration inside the cell, they may result in the movement of this solute or solution from the inside to the outside. So what happens? This may result in cellular shriveling and eventual death of the cell as a result of shrinkage may occur. Actually, this peptidoglycan is the main stress-bearing layer of the bacterial cell wall. And the peptidoglycan also gives the bacterium its shape. The shape is given by peptidoglycan. How this protoplast and sphaeroplast generates? It can be generated both in the laboratory as well as in the nature. In the laboratory, it is generated by weakening the peptidoglycan network in the cell wall. Whenever the peptidoglycan network in the cell wall is weakened by any of the forces, it may generate protoplast and sphaeroplast. Now, what is a protoplast and sphaeroplast? The term protoplast refers to spherical shape, or spherical structure, which are assumed by gram-positive bacteria, whereas sphaeroplast, which may refers to the spherical shaped structures which are assumed from the gram-negative bacteria. Mainly the difference between the protoplast and sphaeroplast which may lies in the presence of a single membrane in the case of protoplast and the presence of double membrane or two membrane, that means the cytoplasmic membrane and the outer membrane of the gram-negative sphaeroplast. So, the sphaeroplast possesses two membranes, whereas protoplast possesses single membrane. Thus, in essence, the protoplast refers to a bacterial sphere that is bounded by a single membrane. It is bounded by a single membrane, and sphaeroplast refers to a sphere that is bounded by two membranes. Next is the preparation. How these forms can be prepared? Both the sphaeroplast and protoplast adopt a spherical shape. Why? Because this shape protects those sphaeroplast and protoplast against hostile environments. However, despite these structural changes, these cells are very susceptible to osmotic stress like osmotic pressure, as the cell wall is, because The cell wall is responsive to environmental ionic concentration difference. So if the cell wall is absent, so how it can withstand those osmotic pressure or any other ionic concentration difference? It is quite difficult for the structures to withstand the osmotic pressures. The bacteria are induced to form the protoplast or sphaeroplast typically by laboratory manipulation. So it can be converted to these forms by laboratory manipulation. But the formation of the structures can also occur naturally. How? Such bacterial forms are called L forms, which we have discussed earlier. So naturally occurring forms are L forms. If they are occurred in the laboratory, then they are called protoplast or sphaeroplast. Examples of the bacterial genera that produce L forms. We have already discussed these portions, including Bacillus, Clostridium, Haemophilus, Pseudomonas, Staphylococcus and Vibrio. These are the bacterial genera that produce L forms. Therefore, when these cells are created within the laboratory, they must be formed with isotonic solutions. When these cells are created in the laboratory, they must be formed with isotonic solution. Why? Because if there is a higher concentration of solution outside, this point we have discussed in the previous slide. Once more I am discussing why these structures are formed within the isotonic solution. Otherwise what happens? Isotonic means outside the cell, inside the cell is in same concentration. If the higher concentration of solution is outside the bacterial cell, what happens? This may result in the swelling and eventual cellular bursting. Whereas whenever the increased concentration, when the concentration is inside in the cell is more, what happens? This may result in cellular shrinkage. So the process of creating protoplast and sphaeroplast must be done in the solution in which ionic composition and the concentration of the fluid outside of the bacteria, it should be same as the inside of the bacteria. That means in the isotonic form or isotonic solution, these forms should be prepared. So these forms are prepared only in the isotonic form. Once the structural support of the peptidoglycan is lost as a result of the preparation of this protoplast and sphaeroplast, when the structural support, that means the peptidoglycan is lost, what happens? The bacteria are unable to control their response to difference in the ionic composition between the bacterial interior as well as bacterial exterior. If these structures are lost, this bacteria should face difficult to control the changes in the ionic composition between the interior of the bacteria as well as exterior. If the inner concentration is greater than the outer ionic concentration, water will flow into the bacterium in an attempt to achieve an ionic balance. The increased volume can be so severe that the bacteria will burst. Similarly, if the inner ionic concentration, if it is less than the exterior, water will exit the bacterium and in an attempt to dilute the surroundings. So what happens? The bacteria can shrivel and it may result in the death of the bacteria. So in the laboratory, we have already discussed that this protoplast and sphaeroplast should be prepared in an isotonic form. Now, how it can be formed? Either through mechanical or enzymatic methods. This we should follow while we are making this sphaeroplast or protoplast. The fungal cells can also form protoplast after chitinase treatment. Chitin is an outer covering of the fungus, and if we are removing this chitin layer using the enzyme chitinase, similar structures are formed like protoplast and sphaeroplast within the fungi. The plant cells also form this protoplast following using the enzyme treatment such as pectinase for the removal of outer layer pectin, cellulase, silanase treatments. But in the gram-positive and gram-negative bacterium are treated with what? It can be either, we can remove this peptidoglycan layer by either treating with wall-digesting lysozymes. In the last class we have discussed about these lysozymes, enzymes which are capable of cleaving the peptidoglycan layer. So this peptidoglycan layers can be within the cell wall can be cleaved using this enzyme. So sphaeroplasts and protoplasts can be generated using lysozymes or wall inhibiting antibiotics. So the antibiotics such as penicillin. Penicillin can be used in order to cleave the outer cell wall structure to form protoplast and sphaeroplast. By exposing the bacteria to an enzyme called lysozyme. Now how this lysozyme action taking place? The interconnecting strands of two particular sugars within the peptidoglycan can be cut. What are the two particular sugars within the peptidoglycan? NAM and NAG, N-acetyl glucosamine and N-acetyl muramic acid are the two sugars which are present within the peptidoglycan. And if the lysozyme, when the bacteria is exposed to this enzyme, these sugars can be cut. When this is done, what happens? The peptidoglycan loses its ability to serve as a mechanical means of support. So the mechanical means of support of this peptidoglycan is lost as a result of the removal of the this sugars when the sugars are cut. This same situation in the yeast, if it occurs in the yeast, what happens? It is slightly different as other components of the yeast cell wall are degraded in order to form protoplasts. In the yeast also this protoplast forms occur, but it may result in the degradation of other components of the cell wall of the yeast also. So the preservation of ionic balance, it is required to ensure that while we are making this, while we are making this protoplast and sphaeroplast from the normal bacteria, using this mechanical methods, we should ensure that the bacterium should not be killed. Otherwise, we can't prepare this type of protoplast and sphaeroplast. That is why we are maintaining these forms in isotonic. We are preparing these forms under isotonic condition. That's why in the hypertonic and hypotonic conditions may result in the death. That's why these forms are prepared in the isotonic condition. So the preservation of ionic balance is required to ensure that bacteria will not be killed during the transformation into either protoplast or sphaeroplast. That is important. The bacterium should not be killed. That's why we are maintaining them in isotonic form. Living protoplast and sphaeroplast. Why we are preparing this living protoplast and sphaeroplast? Because they are valuable research tool. The membrane walls that are the protoplast or sphaeroplast can be induced to fuse more easily with similar structures as well as with eukaryotic cells. So this protoplast and sphaeroplast can be induced. They can be induced to fuse more easily with the similar structures. They can fuse easily with similar structures also with the eukaryotic cell. They are capable of fusing. That I will tell in the later slides. And also this can be used in the transfer of genetic material between the two cells. As well, the sequential manufacture of sphaeroplast and protoplast in gram-negative bacteria allows for the selective release of contents of protoplasm.